Good morning, guys, and welcome back to Excelsior Academy. So today we'll be doing about the cell signaling. So in the previous lecture, we discussed about the GPCR, right? Now we are going to talk about tyrosine kinases and their signaling. So it is a little bit similar to your GPCR and the important one. Now, as we saw about the GPCR, in the case of the tyrosine kinases, they are mainly involved like for their drug therapy also. Okay. So mainly your cancer related any drug is being in the market. It is there in the market. Mainly they target the tyrosine kinase only. Okay. We'll see how and why it is easy for the drugs to target tyrosine kinases. So tyrosine kinases, they phosphorylates the substrate proteins on the tyrosine residues. So we know kinase means adding the phosphate group. Now tyrosine means at the tyrosine, it will be adding. So tyrosine has got another OH group to which it will add the phosphate group, right? Now, in the case of the tyrosine kinase, the signaling pathways are involved in the control of animal cell growth and differentiation. Now, because of which the drugs are mainly made against the tyrosine kinase domain, okay, especially for the cancer chemotherapy. Indeed, the first tyrosine kinase was discovered during the studies of oncogenic protein called Rho sarcoma virus, which directly linking tyrosine phosphorylation to the abnormal growth of the cancer cells. So this was the first study. Many of the drugs currently being developed as new cancer treatments are inhibitors of tyrosine kinases or the downstream signaling pathways. So like during my work also, when I am carrying out all these clinical trials, I go through the clinical trials to understand whether that particular cancer, how that particular cancer is being treated. So many a times I see that the drugs which is being used in the clinical trials, okay, and it is mainly it is against the tyrosine kinase, and or otherwise you will have which are tyrosine kinase inhibitor. This is tyrosine kinase inhibitor, okay. So there are certain cancer cells which are resistant to this. So then you have drugs which are resistant to your tyrosine kinase inhibitor. So iske upar naya drug is made. Okay. So mainly till now what I've seen is that they always target the tyrosine kinase. Now, receptor tyrosine kinases. Maybe in the next lecture, I'll show you like how the clinical trials looks like and how to understand. Okay, and I would suggest to everyone, if you want to make a like career in cancer related, okay, anything in cancer related, or sorry, or you want to have a like be involved in the clinical trials. So clinical trials doesn't mean that only working with the cancer. They do have clinical trials for the diabetes, for the other disease also, but there are like many institutes which there are many institutes and organizations which are taking the clinical trials in India. So if you want to be part of it, like if you think that you can contribute over there, okay, so it is not as such research, but half research and half interacting with the patients over there. So if you want to be interested over there, you can do the courses which is related to the clinical research. Okay, so you can try doing that also, because that is like new market now after this pandemic of the COVID. It has really become heightened, and many fundings was also there by the government also, and as well as by the private sector also. So that is now new market. Apart from the pharma, apart from being a doctor or engineer, even you can be a CRO. What is CRO? Clinical research organizer. Okay, so you can be a part of it also. Okay. Now coming back to the tyrosine kinases. So the receptor tyrosine kinase, it includes the receptor for most of the polypeptide growth factors, establishing tyrosine phosphorylation as a key signaling mechanism in response to cells to growth factor stimulation. So what we are saying here is that, see, if this is your tyrosine kinase domain, okay, either you can have a single or you can have a dimer. Now what happens is that they come together and form a ligand binding domain. Okay, now this receptor can be for the EGF, or it can be of NGF, PDGF, etc. So when these ligand comes in binds, these two come together and they dimerizes. Okay, so all these receptors, they share a common structural organization. So we have got the N terminal in the extracellular binding domain, a single alpha helical domain, and the C terminal domain. Now in the C terminal domain only, you have got the tyrosine kinase domain. Okay, now one more thing. What will happen if the strands comes together, okay, and the ligand binding domain is being removed? So does it mean that the receptor has become constitutively active or inactive? Did you get the question here? If I remove this portion which dimerizes in order to bind to the ligand, let us say that this portion has been removed. Does it make your tyrosine kinase receptor active or inactive forever? Yes, what will be the answer? Rishita? Inactive? Sure. Yeah. So it is inactive if it is being removed at the starting. Okay. But if suppose they come together, let's say that they come together and dimerizes. Okay. And after that you remove this portion, then it becomes constitutively active. Okay. Because see, why I'm saying is when they come together, this kinase domain will phosphorylate here and this will phosphorylate over here. So when they come together, they actually auto-phosphorylate each other. And that's how they give the signal at the downstream region. Okay. But if they are like, separate, so ये phosphorylation नहीं होगा. So that time it is inactive. Okay. But if they come together and they are in close vicinity, that's the time when it becomes active. And uske baad whether this portion is there or not, it doesn't matter. Then it will remain active only. Okay. Now the binding of the ligands or any other growth factors to the extracellular domains of these receptors activates the cytosolic kinase domain, resulting in phosphorylation of both the receptors by themselves. So that's why it is autophosphorylation. The first step in signaling from most receptor tyrosine kinase is ligand-induced receptor dimerization. So that's why I was saying, so whenever these growth factors like PDGF or NGF, they come together, they bind to the receptor and they dimerizes. And after that only the autophosphorylation takes place. So when I'm saying that it's a dimerization, obviously the growth factors which is also binding, okay, they need to be also a dimer. Clear? Now, other growth factors such as EGF, they are monomers, but leads to receptor dimerization as a result of inducing conformational change that promote protein-protein interactions between different receptor polypeptides. So here we have, see, receptor dimerization, this is how the growth factor will go and bind, and when they come together, they autophosphorylate. So what is happening over here is that ये जो tyrosine kinase है, okay, this is going to phosphorylate this at tyrosine residues, and here it is going to phosphorylate this at the tyrosine residues. Okay, is this much is clear? So this is known as autophosphorylation. Now, ligand-induced dimerization then leads to autophosphorylation of the receptor. Okay, such autophosphorylation plays two roles in signaling from these receptors. Pehla, phosphorylation of tyrosine residues within the catalytic domain increases protein kinase activity. So now you have made your tyrosine kinase receptor more active. Second, phosphorylation of tyrosine residues outside of the catalytic domain creates specific binding sites for additional proteins that transmit intracellular signals downstream of the activated receptor. So what does it mean over here? What they want to say is that, see, you have it over here, and if you notice, the phosphate group has been added over here, right? It is towards the outside. Now what happens is that the proteins will come and bind over here. Okay. Now they can only bind after the phosphorylation, because at the active site, this phosphate group is needed. So these proteins will come and bind like this. Okay, so it's actually making a correct active site for your other proteins to come and bind. So the association of these downstream signaling molecules with receptor tyrosine kinase is mediated by protein domains which will bind to specific phosphotyrosine containing peptides. Okay, so it means at the active site, the phosphate group is needed. For example, SH2 domains, which stands for SRC homology 2. They were initially recognized in tyrosine kinases related to SRC, the oncogenic protein of through sarcoma virus. So this is how. So once the growth factor comes, binds to the tyrosine kinase receptor, it dimerizes it, right? Now the tyrosine kinase is going to autophosphorylate the each other. Then what do we see? Now that becomes the activated site for your SH2 domain to come and bind. So these are nothing but your downstream signaling molecules. Okay, so they come and bind over here. Now, अब क्या होगा इसके बाद this is going to activate further the downstream proteins, and that's how the signal is being carried forward. Okay, is this much is clear to everyone? Chandni?
Yes.
Okay. Now coming to the non-receptor tyrosine kinases. So you see, as you see in this case also, it is hugely different from your GPCR, right? So GPCR में कैसे था? The any growth factor or the ligand comes and binds to the GPCR. What happens to the GPCR? Conformational change is there. Then you have got your beta, gamma, and alpha together, right? So यहाँ पे ये downstream था, which was taking or carrying out the signal. If alpha gets activated, it is going to activate your adenylyl cyclase, and then you have got the increased concentration of CMP. Then CMP have its all regulatory effects. Whereas in the case of TK, that is tyrosine kinase receptor, the ligand binds, dimerization happens, autophosphorylation happens, and then only the downstream molecules binds to it. Okay, so this is how. Please try to remember also. Now coming to the non-receptor tyrosine kinases. Rather than possessing intrinsic enzymatic activity, many receptors act by stimulating the intracellular tyrosine kinase, with which they are non-covalently associated. So here we don't have the tyrosine kinase domain present. Okay, this will be activated by the other kinases. So example includes the members of the cytokine receptor superfamily, which encompasses the receptors of most cytokines and for the peptide hormones. So they do contain N-terminal at the extracellular level, then again single transmembrane alpha helices and T-terminal cytosolic domain. So what do we see here? It is similar to your tyrosine receptor, okay, tyrosine kinase receptor. The difference is that here it cannot autophosphorylate. Okay, they don't have the internal kinase domain. They need other proteins, okay, which come and bind to the cytosolic part of your receptor, and then they do phosphorylation. Did you get it? Did you get the difference over here, guys? So it is not the cytosolic tyrosine kinase domain. Okay, they don't have it in the case of the non-receptor. So the other proteins, okay, which are present in the cytosol, they come and bind to the cytosolic site, and then they go for the phosphorylation. Okay. However, the cytosolic domains of the cytokine receptors are devoid of any catalytic activity. Instead, the cytokine receptors function in association with the non-receptor tyrosine kinases and are activated as a result of ligand binding. So what is happening? The first the ligand is coming, in this case it is cytokine, right? So it is cytokine is coming and it is binding to its receptor. Here the cross phosphorylation of the associated non-receptor tyrosine kinases is happening. So then what will happen once they get phosphorylated? Okay, then again the SH2 domains will come because they just recognize the phosphate attach, the cytosolic part of the receptors. Did you get the difference between the receptor kinases and the non-receptor kinases? Here, the different proteins will come and bind, okay, and then they will do the phosphorylation. For example, the kinases which are associated with cytokine receptors include the members of the genus kinase family, which consists of 4 related non-receptor tyrosine kinase. So this is related to your JAK-STAT pathway. Okay, so the JAK genus kinase, they come and bind to the non-receptor kinases of the cytokine. So they are the cytokine receptors. Cytokine comes and binds, dimerages, genus kinase comes and then they phosphorylate. Now after that, the STAT protein comes. Okay, they are going and binding over there. They get activated. That is, वहाँ पे आपका phosphate group transfer होगा. Okay, and then they dimerages. So the key target of the JAK kinases are the STAT proteins. What are STAT? Signal transducers and the activators of the transcription. So they are inactive in unstimulated cells where they are localized to the cytoplasm. So what do we see here? So जैसे ही cytokine आएगा, non-receptor tyrosine kinase को bind करेगा, JAK will come, it will phosphorylate and makes it active. Then the STAT will come. So these STAT will get phosphorylated and they will combine together to form the dimer, and then they will carry out the signal which is required for your transcription, okay, of those particular genes. So after dimerization, okay, the STAT proteins which are phosphorylated by the JAK, they go into the nucleus. Okay, then they're translocated into the nucleus. The STAT, okay, they are the transcription factors serving as a direct links between the cytokine receptors on the cell surface as well as the regulation of gene expression in the nucleus. Is it clear, guys, to everyone? Tanvi, is it clear?
Ma'am, can you repeat this part?
Okay.
This, yeah.
So here what is happening is that you have, this is about the non-receptor tyrosine kinase, okay? Non-receptor tyrosine kinase. So here this is the example of cytokine receptor. So this is your cytokine receptor it is, okay? Now the cytokine is coming, binding to the cytokine receptor, dimerization happens, which recruits your JAK. JAK is your genus kinase. Okay, it is going to phosphorylate. Now, after phosphorylation, it is recruiting your STAT proteins. Okay, STAT proteins gets phosphorylated by the by binding over here, so they get phosphorylated. Now, after phosphorylation, they forms the dimer. Now, this dimer, okay, it will be translocated to the nucleus where it is going to act as a transcription factor and going to stimulate the transcription of their target genes, by which cytokine is responsible for certain gene expression. So that will be now carried forward. So transcription होगा उन particular genes का. Okay, so what are those genes responsible for? They are responsible for the which is result. So whenever the cytokines are released, so what kind of genes are required? So those will get expressed. Okay. Now, another example of non-receptor tyrosine kinase belongs to SRC family. This is about the root sarcoma virus wala, okay, those proteins. So members of the SRC family play important role in signaling downstream of cytokinin receptors, that is antigen receptors on the B and T lymphocytes. Okay, so they are involved in cell-cell as well as cell matrix interactions. For example, so whenever we just now completed about cell-cell and the cell matrix interaction, so integrins play important role if you remember, right? So integrins, they play important role in maintaining these interactions. Integrins also serve as receptors that activate intracellular signaling pathways, thereby controlling the cell movement and the other aspects of the cell behavior. Okay, so let's see how it is going to regulate about it. Now, So the one mode of signaling downstream of integrins involves activation of non-receptor tyrosine kinase called FAK, that is focal adhesion kinase. So you know, if you remember in the focal adhesion, how integrins are helping in binding to the extracellular matrix, right? So that it can make attachment, it can make connections with the extracellular matrix. So can I say that you guys remember? Do you remember how the focal adhesion looks like? Yes. Do you remember the proteins which are involved? See, if suppose this is your cell and here the extracellular matrix is there. Okay, so I said, right, the integrins will be present like this. So integrins of the cell membrane, okay, they're exposed out and they make connections with the extracellular matrix over here with the collagen, with the fibronectin and what all is present over here, right? Then this integrin is made up of alpha and beta subunit. They are joined with the talin and vinculin, if you remember, to the actin fibers like this. And each actin fibers are connected with the help of alpha actinin, if you remember. And this, with the help of the talin and vinculin, they are connected to the integrins molecules. Okay, so guys, don't forget all this thing, it is important, and that's how the focal adhesion is made between the cell membrane as well as the extracellular matrix. Okay, coming back over here. So for this we need FAK, focal adhesion kinase. So it is localized to focal adhesion and rapidly becomes tyrosine phosphorylated following the binding of integrins with the extracellular matrix components. So the moment your integrins are making connections with the extracellular matrix, the FAK is getting phosphorylated. Okay. Now, the activation of FAK involves autophosphorylation, which is induced by clustering of integrins bound to the extracellular matrix. So, what is happening? Integrins are making connections with the extracellular matrix. The FAK is getting phosphorylated. So, autophosphorylation of FAK creates docking sites for the signaling molecules containing SH2 domain, including the members of the SRC. So, again, so what is happening? You are going to have the phosphate group like this, and SH2 domain will come and bind like this. Okay. So interaction between cells as well as between cells and the matrix play critical roles in multicellular organisms. Here, see, integrins are there. As you can see, this is the plasma membrane, integrins are there, and it is making connections with the extracellular matrix. Okay, the moment it makes a connection, the FAK which is associated with the integrins, they get activated and they autophosphorylate. They are focal adhesion kinase. So once this is being done, what happens? The SRC. So SRC is your protein, is it has got the SH2 domain. Okay, so it is this SH2 domain which actually binds to the phosphorylated portion. Okay, now what is happening? When SRC binds, then further downstream molecules will get activated by the SRC function. Is this much is clear? How the integrin molecules are in connections with the extracellular matrix, then how FAK is getting autophosphorylated so that the SH2 domain of the SRC can bind to it. Is it clear to everyone? Akshaya?
Yes, ma'am.
Okay. Now coming to the another pathway, that is MAP kinase pathway. So let's little bit summarize what we have just discussed now. So in the previous lecture, we have discussed about the GPCR, okay, tyrosine kinase, both receptor and the non-receptor. So these three pathways we have discussed now. Now the fourth one is the MAP kinase pathways. Okay. Okay, so MAP kinase signaling is one of the major pathways of signal transduction which is activated downstream of both receptor and the non-receptor tyrosine kinase. So this is downstream of the tyrosine kinases. The MAP kinase is downstream. Okay, so it refers to a cascade of protein kinases which are highly conserved in evolution and play central roles in signal transduction in all the eukaryotic cells ranging from yeast to humans. Okay, now MAP kinase, they are the central elements in the pathway. They belong to serine threonine kinases, mitogen activated protein kinase. It gets activated in response to variety of growth factors and other signaling molecules. Okay, so this and all you can see that they're involved in the mating, cell shapes, sporulation, present in many higher organisms. And it is characterized that it is belonging to the ERK family also, the MAP kinase. Okay. So जब भी हम बोलेंगे ना, like in the exam whenever they mention, they always mention like this: map ERK kinase pathway. Okay, or map ERK pathway, we mentioned like this. And like I can show you also, like in the clinical trials they also mentioned that the drugs are your map ERK MEK, ये इसके नीचे आएगा, ठीक है. They are the inhibitors, so it means what, they are going to inhibit this pathway, यानी कि MAP kinase pathway वो inhibit करेगा, ठीक है. So first what we saw is the GPCR on the top, then we saw tyrosine kinase. इनके नीचे आता है आपका MAP kinase pathway. Okay, MAP के नीचे ERK के नीचे MEK. So we'll see how these are actually getting affected. Okay, guys, please join back. Okay, so as I was talking about the map, so in many higher eukaryotes it is present. Okay, so they are the ubiquitous regulators of the cell growth and differentiation. Now the MAP kinase, which was first characterized, okay, so that was the ERK one, extracellular signal regulated kinase family. So your MAP, which is belonging to your ERK family, okay, and in this also the RAS proteins are playing important role. So please remember that. So MAP kinase is your mitogen activated protein kinase. Now, especially they are involved in your, during the mitosis. We'll come to that mitosis. So they are belonging to your ERK family. Okay, and in that also the RAS proteins are also very important. Now, keeping these things in the mind, we'll talk about this entire pathway, which is important. Okay, so we know what is RAS. We know that it is important. It induces proliferation of normal mammalian cells. If suppose something happens to the RAS, then obviously it is going to lead to the cancers. Okay. Now, what is RAS? We know that it is a G protein. It is a guanine nucleotide binding protein, and it functions analogous to the alpha subunits of the G proteins. यानी कि क्या, RAS GTP is active, RAS GDP is inactive. Okay. Now, RAS activation is mediated by guanine exchange factor, which will stimulate the release of bound GDP in exchange of GTP. Okay. Activity of the RAS GTP complex then terminated by GTP hydrolysis, which we know, which is then stimulated by GTPase activating proteins gaps. Now this is important. Again, this is really very simple. जो हमने last time discuss किया था in the case of your G protein, right? So there also guanine exchange proteins were required, factors were required, and there also guanine activating. GTPase activating proteins are required. Same way in this case also. अगर आपके पास RAS GTP है, GDP hai, it is inactive, then guanine exchange factor will come and GTP लाएगा instead of GDP. So this becomes active. Okay. Now hydrolysis लाना है इसका. So this will be brought about or stimulated by GTPase activating proteins. So this is how the RAS proteins or any other G proteins are regulated. Okay. So that is all about the RAS proteins. Same thing written over here. Okay. Now, I'll go to the figure directly. That will give more understanding. Okay. See, this is the pathway. So, जो हम बोलते हैं ना, oh shit, current gone. Okay. So, this is the pathway. Okay, this is important, and the same thing is written in the back also. Like after discussion, we can move back over there. So once the growth factor comes and bind to the tyrosine kinase receptor, what happens then? the phosphorylation, and then SH2 domain comes. Okay, now this SH2 domain is gonna activate your Jeff. So Jeff kya karega? Guanine exchange factor, it is going to make your RAS active. So from RAS GTP, it is going to make RAS GTP. Okay, now RAS GTP once activated, it is going to activate your RAF. Okay, I'm going very slow. Okay, now this is going to activate your RAF protein. Now what is RAF? Again, RAF is as a protein kinase. It's a kinase. So it is going to phosphorylate your MEK. Okay, MEK. Then that is going to activate your ERK. Then ERK is going to activate the other nuclear and the cytoplasmic target proteins. So this is your MAP pathway, or the MAP ERK MERK pathway. Clear? Simple. ग्रोथ फैक्टर बाइंड हुआ, SH2 डोमेन बाइंड हुआ आपके टायरोसिन रिसेप्टर में, ठीक है. जेफ एक्टिवेट्स योर RAS फ्रॉम GDP to GTP. It activates RAF, MEK, ERK. It is going to activate matlab they all are kinase, so they are going to phosphorylate the downstream proteins over here. Okay, now keeping these things in the mind, let's see what then the theory, if suppose I miss anything. Okay, so this we have discussed. Okay, so you know what is ERK now. ERK is your extracellular signal regulated kinase. MEK क्या हो गया? आपका माइटोजन related protein हो गया है. Okay, so this we have covered. Okay, so this localizes JEFs to the plasma membrane where they're able to interact with the RAS proteins, and then you can activate from RAS को GDP के जगह आपने GTP add कर दिया है. Okay, simple. Now, in its active GTP form, RAS interacts with number of effector proteins like RAF. RAF is again another serine threonine kinase. Okay, then it is going to phosphorylate another protein kinase called MEK. MEK is your MAP kinase or ERK kinase. Now, after that MAC or the MERK, it is going to phosphorylate the other proteins. Okay, so what we have seen is that in this entire process, okay, entire pathway, it is just adding the phosphate group. So okay, can I say that this pathway is clear to everyone? It's really very simple. Do you want me to explain once again? Krishita, is it clear?
Yes, ma'am.
Yeah, it's really very simple. It's just that you need to remember which one comes when. Okay, so when we say that map, ERK, or MEG pathway, whatever it is, you need to just remember that after this SH2 domain के बाद से JEF, RAS, RAF, MEG, ERK, बस इतना आपको याद रखना है. ठीक है? Okay. A fraction of activated ERK translocates to the nucleus where it is going to regulate the transcription factors by the phosphorylation. Then it is notable that a primary response to growth factor stimulation is rapid transcriptional induction of a family of 100 genes called the immediate early genes. Now, why immediate early genes? What are we discussing? We are discussing about the example in the case of the Rho sarcoma virus. So in the case of virus, you know that there are immediate early genes, late genes are there, right? So this is just the example where it is going to activate, or it is going to induce the transcription of these immediate early genes. Okay, now the induction of these immediate early gene is mediated by regulatory sequence called the serum response element. Now this is recognized by complex of transcription factor including serum response factor and ALK1. Okay, now what is happening? So हमने देखा अभी like from Jeff RAS activate हुआ, RAS के बाद RAF activate हुआ, फिर MERK हुआ, फिर ERK हुआ. Now this phosphorylated ERK will come into the nucleus. Now this is going to activate your ALK1 and SRF, which will go and bind to your SRE, that is serum response element, and it is going to induce the transcription of the immediate early genes. Is it clear? This is the second part of the pathway where we are talking about the immediate early gene induction. Able to follow? So what are ALK1 and SRF? They are the transcription factors responsible for binding to serum response element, which is going to induce the immediate early genes का transcription. ठीक है? Same what we had just discussed. So ERK phosphorylates and activates ELK1, providing a direct link between the ERK family of MAP kinases and the immediate early gene induction. So immediate early genes themselves encode transcription factors. So their induction response to growth factor stimulation leads to altered expression of battery of other downstream genes also called secondary response genes. Okay. So the moment immediately genes are being induced, the later it is going to also induce the expression of the other downstream proteins required. So that's all, guys. Any doubts in today's pathway, tyrosine kinase? Yes. Let's summarize. Okay, let's summarize. So today we discuss about the tyrosine kinase. So in the case of the receptor tyrosine kinase, what we have seen that whenever whenever ligand comes, it dimerizes, right? And after dimerization, the cytosolic domain, it contains your tyrosine kinase domain. So they are going to autophosphorylate. So once they are phosphorylated, the proteins, like for example SRC, they come and bind. Now the binding of this portion, okay, this should have your SH2 domain. This is like by default, only that can bind. This is in the case of the receptor. Now if we have got the non-receptor tyrosine kinase, then what is happening in this case? The ligand is coming and binding. Okay, now the dimer is found, but here the another protein, okay, from the cytosol will come and bind over here. They are going to do the phosphorylation. Again, to this phosphorylated tyrosine kinase domain, the SH2 protein will come and bind. Clear? Right? This is what we have discussed, right? Now, later what happens? If suppose let's talk about the MAP kinase pathway. So in the MAP kinase pathways, what is happening? After getting activated, the SH2 domain is there, right? Let's say that here the phosphate group is there, SH2 domain ने यहाँ पे इसको bind किया of SRC protein, okay, SRC protein. Now this activates your JEF. JEF क्या करेगा? Guanine exchange factor. It is going to activate your RAS GDP से उसको बनाएगा RAS GTP. Okay, this is going to activate your RAF kinase. RAF is going to activate your MEK. That is by phosphorylating. This is also by phosphorylating. MEK is going to activate your ERK. That is again phosphorylated. This will go inside the nucleus now. Inside the nucleus, for example, if you're talking about the induction of immediate early genes, inside the nucleus, ERK-P is going to activate your ELK1 and दूसरा क्या था आपका? Serum response factor, right? So this will be activated. This will go and bind to your serum response element and induce the transcription of the immediate early genes in the case of the Rosarcoma virus. Okay. So any doubts? Is it clear?
Yes, ma'am.
Yeah. Okay.
Yes, ma'am.
Good. So I'll be stopping here now.